Hello everyone, and welcome to today's webinar, organised by The Pathologist and VizGen. I'm Georgia Hume, Associate Editor of The Pathologist, and I'm super excited to be hosting today's talk on how spatial analysis of human lung cancer has revealed organised immune hubs rich for stem-like CD8 T cells. I'm excited to introduce Nir Hacohen, who, among other things, is David P. Ryan Professor of Medicine at Harvard Medical School and Director of the Center for Cell Circuits at MIT in Harvard. He will be discussing his team's recent work and its heavy relevance to patient immunotherapy outcomes. At the end of the presentation, Nir will be taking questions from audience members, so please feel free to submit your questions during the course of the talk. It is now my pleasure to hand the reins over to Nir, who will moderate the session.
Thank you for the introduction, and I'm very happy to be speaking here today. What I'm going to talk about today is how the immune response is organized in space, in particular in tumors. And just as a quick introduction to how the work we do happens, we basically take a systems approach to immunology across cancer, autoimmunity, severe infections, asking in a systematic way what are the cell types, the factors, the pathways that are associated with particular conditions, in particular the control of cancer by the immune system, the development and progression of autoimmunity, the progression of infections toward sepsis. And we use both single cell and spatial methods to understand what's happening in these systems, and also to do causal studies to understand what controls these processes using CRISPR screens, both in vitro and in vivo. So what I'm going to talk about today is really the factors that drive immunity in cancer. There are many factors that can drive immunity, including tumor antigens and the targets. And we've studied mutated antigens for many years. There are many cell types and interactions that contribute to immunity, and that's what we'll talk about today. And there are many other triggers and inputs and features that affect immunity against cancer, which we won't talk about today. So we'll focus today about cell-cell interactions, the structures that form between cells and the niches they create. And so the main question we want to address today is, what is the spatial organization of the immune response in tissues in cancers? Lymphoid organs, like lymph nodes, are very organized tissues with different immune cell types at different locations. But is it true then that a cancer, which is a more haphazard organization in the sense that the tumor itself is not an organized tissue, would the immune cells just be coming in and floating all around and not have any particular organization? And so we don't really understand that very well. So what I'll tell you about today is our efforts to understand this question, starting first looking at what are the cells and the cell types and the gene programs within tumors. Can we use that information to infer the possibility of a multicellular interaction network of cells talking to each other? And if we can, how do malignant cells in the tumor and the immune cells participate in these hubs or organized areas of spatially coordinated cells? Can we potentially impact genes in these spatial hubs with therapies? And do these hubs generalize to other tumors, predict response to immunotherapy? So we'll start with the main question we addressed in the first study that I'll talk about today, which is what mechanisms underlie this dramatic difference in the response of mismatch repair deficient versus mismatch repair proficient tumors. So in immunotherapy with checkpoint therapy, in particular anti-PD-1 therapy, there's a well-known finding, which is the patients who have tumors with deficiencies in mismatch repair, meaning that they have more DNA damage and more mutations, respond very well and have great survival curves in response to immunotherapy with greater than 50% response rate of those tumors. On the other hand, mismatch repair proficient tumors of the same type, in this case colon cancer, what we'll be talking about, there's no response. In fact, almost 0% response rate to tumor immunotherapy, anti-PD-1 therapy. So we wanted to understand really the difference in the immune response between these two conditions. And so we collected a set of 62 patient samples with untreated colorectal cancer so that we could see what is the baseline immune milieu and immune programs that are on in these patients. And we collected around the same number of mismatch repair deficient MMRD and mismatch repair proficient patients. There are a lot fewer of these, so we had to collect a lot more, wait a lot longer to collect these patients, but we wanted to get a fairly equal number so we could compare them. And we did single cell RNA-seq of freshly dissociated tumors in adjacent normal tissue and collected about 1 million cells. And what I'll tell you about today is 400,000 cells from these patients that separate into 7 cell lineages and 87 clusters. In addition to cluster analysis that identifies cell types that are different, we also wanted to look at gene programs, because not only do cells separate from each other, but there are sets of genes that work together. So for example, a couple of genes may be activated and induced at the same time, and that would constitute a program of genes that are co-regulated. And so we used an approach called non-negative matrix factorization that allows us to identify these programs. For example, here we see the proliferation program. Almost any cell type can undergo proliferation, but that program does not need to be on in all cells. So only a fraction of a cell or a particular cluster may be in proliferating state. Or the interferon stimulated gene program that gets turned on in particular cells within a cluster. So we really want to understand these gene programs because they can go across cell types and are kind of orthogonal in the thinking to cell clustering. And what we were able to do is to then use these cell programs to ask which cell programs are correlated in their level of activity across cells. So for example, imagine a tumor specimen, 1, 2, and 3 different tumor specimens from different people. And you ask the question, are programs in different cells, let's say program 2 in one cell type and program 3 in another, correlated to each other, such that as you go from tumor to tumor, if you go higher amounts of the P2 program in these cells, you get higher amounts of P3. So is there a correlation between them? And when we create a correlation diagram here shown below between all the programs versus all the programs, you see that you get these blocks, which we call hubs. And I'll talk about them in a minute in more detail. But the idea is that these are programs that are interacting. And so that way that programs can interact is if the cells are next to each other and the cells communicate with each other. One program turns on the other and forms a positive feedback loop, or it's possible that an external signal is inducing the programs in both cells and thus coordinating them. So we found in MMRD tumors 7 of these coordinated hubs and 9 such hubs in the MMRP tumors. You can see that there's one hub particularly that is very, very large that's present in the MMRD, and actually we don't see that in the MMRP. And that's the one for today that we'll focus on, although there are many other interesting coordinated hubs. Zooming in on this hub, you can see the structure of the hub, and I just want to point out a few key programs that are correlated to each other. In T cells, we found that a CXCL13 T cell program, which I'll mention and explain in a moment what it is, an interferon gamma MHC2 T cell program, and a T cell proliferation program are present in this hub. In malignant cells, we see a program, and P stands for program, epi for epithelial, and this is just the number of the program. That MHC ISG program that's getting activated, and we'll talk about what that means. And then in myeloid cells, we see interferon stimulated gene programs in macrophages, and also we see dendritic cells. So these programs are correlated to each other, meaning that they are going up or down together with each other as you go from patient to patient across the 60 or so patients. And these programs also tend to be higher in mismatch repair deficient patient compared to proficient patients. And you can see that healthy or normal adjacent tissue really doesn't have these programs at all. Mismatch repair proficient only occasionally has them, and most of the mismatch repair deficient patient tumors have these programs. So what's the CXCL13 positive T cell program? Well, when we stain for CXCL13, which is a chemokine that would usually bring in B cells, for example, we find that the CXCL13 positive T cells are really present throughout the tumor. Traditionally, people think of CXCL13 as present in lymphoid structures. In fact, we do see it there, but it's not made by T cells in those lymphoid structures. It's made by other cell types. So CXCL13 marks T cells that are present throughout the tumor. But what's special about these cells? Well, in a study we published with Kathy Wu a couple of years ago, by sequencing T cell receptors in melanoma biopsies, we sequenced many TCRs. We clustered them into subsets across the single cell RNA-seq. You can see different subtypes of cells, memory cells, activated cells, proliferating T cells. We cloned the 172 top TCRs across all these T cell subsets. Those were clonal expansion, and tested whether these TCRs are tumor reactive or reactive to other antigens such as viruses. And when we did that, we found that the melanoma associated antigen reactive TCRs and the neoantigen reactive TCRs are basically localized in particular clusters of T cells, while virus reactive TCRs to common viruses such as EBV and CMV are in a different part of the space of T cell subtypes. And when we look to see what are the top markers of tumor reactive TCRs or tumor reactive T cells, CXCL13 was the top marker. So the program that we see going back here as one of the programs in this hub is indeed a T cell program of T cells that are reactive to the tumor. Now, when we look at the connectivity of the programs within each of these, within MMRD versus MMRP, so these, the thickness of these lines indicates the level of correlation between the programs across tumor samples. As I mentioned before, this is just another way of visualizing that correlation map as a graph. You can see that the graph is highly connected in MMRD, but in MMRP, these programs are really not correlated to each other. Maybe some small bits, but the whole hub is not there. And so you can see basically that this hub forms that correlation structure in MMRD, but not in MMRP. So what's in this hub? Well, we said the CXCL13 positive T cell program is there. There's also the ISG program, which we see in malignant cells, in macrophages, and in dendritic cells. And this ISG program is interesting because these CXCL13 positive T cells produce interferon gamma, which is the cytokine that in fact induces these ISG programs. And as part of these ISG programs, these cells also make chemokines, CXCL9, 10, and 11, which act on another protein on these CXCL13 positive T cells, CXCR3, which is the receptor for these chemokines. So what you get here is a positive feedback loop wherein the T cells that enter the tissue that are tumor reactive stimulate the cells to produce chemokines that bring in more of these T cells. And so you essentially get a coordinated concentration of the T cells in one location. And we believe that this is the way the immune system functions to eliminate a set of targets. So if you have a tumor with billions of cells, in order to eliminate those systematically, you need to have a positive feedback loop that brings in enough T cells that really eliminates and clears out the tumor. So we believe we identified here the positive feedback loop in this hub. Now, we predicted this hub would be spatially localized. So to test that, we then stained for the key markers, CXCL13, which marks the T cells, interferon gamma, which marks the cytokine that is produced by these T cells, CD3, which is a T cell marker, CXCL10-11, which are the chemokines that are made by the other cells that bring in the T cells, and then a marker of the tumor itself. So you can see here that what is going on, if you look from a high level, is you see these regions that are bright, kind of like cities in the night from a satellite photo. And what these are essentially the spatial hubs that are physically manifesting what I told you as the correlation between programs. So these cells really are talking to each other in local regions. You can then zoom into one of these regions and look more closely, and you can see all the components of the system. This is done, I should say, by RNA in situ hybridization, which allows you to stain for RNAs for genes that would not usually be possible to stain by proteins, such as chemokines, which are secreted and don't form particularly strong signals at the protein level. So this allows us to see the T cells as CXCL13, the malignant cells and the non-malignant cells that express these chemokines that bring in the T cells, and so forth. So essentially, each of these little hubs is the manifestation of the predicted correlation. Now, we wanted to ask the question, or we were interested in the question of what targeted therapy does to immune responses. And so, as many people know, BRAF and MEK are common mutated proteins in genes in cancer. For example, in colorectal cancer, there's a set of patients that have BRAF mutations, and some of these patients have this pathway activated. And so by using a inhibitor drug to this pathway and combining that with anti-PD-1 treatment, we were able to ask the question, and this is in a clinical trial run by Ryan Corcoran at MGH, we were able to ask the question, what changes are there in the expression of genes in the malignant cells with therapy? And what you can see is that in patients who had longer progression-free survival, some of the main proteins that are upregulated are ISGs, and in particular CXCL10 and 11 and 9. The chemokines that I mentioned to you before are the ones that bring in the T cells, whereas in those folks who did not do as well, these were not upregulated significantly. And interestingly, when we took organoids that we made from these tumors from these patients, we found that even intrinsically, if you just add the BRAF MEK inhibitors, nothing else, to these organoids in culture, no immune cells, nothing else there, that induces the CXCL9, 10, and 11. And when you add even stronger inhibitors of this pathway, you get even higher induction of these genes. Now, we don't know mechanistically yet how the BRAF-MEK pathway interacts with the pathway that drives these, but it is a very important observation because what we find in these patients is that there's a much higher response to therapy when we combine BRAF-MEK with anti-PD-1, increasing the therapeutic efficacy of both and synergizing. So we believe that this is a potential way in which targeted therapy and immunotherapy may combine by helping to induce the genes that are relevant to basically organize these hubs. So another question we asked is, do these hubs generalize to other cancers? What I've shown you is colon cancer. So now we move to lung cancer. And the reason we move to lung cancer is that most colon cancer patients are not treated with anti-PD-1 therapy because the mismatch repair deficient patients are rare, and the mismatch repair proficient patients, the therapy that I just showed you, the BRAF-MEK combination with anti-PD-1, has not yet been approved to be used. At some point when that's approved, I think we will see a lot more patients treated. But lung cancer, almost every patient with non-small cell lung cancer is treated with anti-PD-1 and often with chemotherapy. So here we collected, together with Justin Gaynor at MGH, a set of 68 patient samples with 20 responder patients and 48 non-responders to anti-PD-1 immunotherapy. And we used the same set of probes I showed you earlier that represent that hub, but we also added a second panel of for hybridization for genes that mark the different T cell states. So we mark here CD8 cells, Ki67 for proliferation, PD-1 for activation of T cells, and TCF7 for activation and exhaustion, and TCF7 for stem cell-like T cells that are really what we believe are the basis for the source for activated T cells in a tumor, and we'll talk about that in a few minutes. So this allowed us to look at the hub that I showed you in colon cancer, plus go in deeper and ask what kind of T cells are present in that hub, not just to say there are activated T cells, but are there other subtypes of T cells? So the first thing is we did find that the hubs are present in non-small cell lung cancer with the same markers that we found in colon cancer. And I should say we've looked at a few cancer subtypes now, and it's present in all cancers that we've seen so far. So how do we identify hubs in these patients? Well, we use CXCL10 and 11 expression, shown here as a sum across the tumor, and find areas, or making a grid where we find essentially areas where there are CXCL10 positive, 11 positive regions. And then if they're adjacent, we combine them and call that a window. And we aggregate them, and we call these immunity hubs. And so these immunity hubs are present throughout the tumor if they are. And what we found was that there's enormous difference in the non-responders versus the responders. This is very clear. These immunity hubs are present only in a small number of non-responders. Most of them have 0, whereas responders, almost all of them have these hubs present, similar to what we saw between MMRD and MMRP, although there we didn't have immunotherapy response data. It was just, we knew that one responds more than the other. Here we actually have the response data. Now, given that we have these T cell markers, we could further subdivide these subclusters. So although all of them may have these markers in them, we asked, do these markers further separate them? And in fact, when we cluster based on all those markers, the hubs that we do find that are CXCL10 11 positive, we find that there's multiple subclusters within them. And when we look at subcluster 3 in particular, we find that subcluster 3 is the one that distinguishes primarily the non-responders and responders. And you can see here, subcluster 3 is the highest difference enriched in responders, both in terms of rank difference and statistical significance. And you can see here the area taken up by cluster 3 spatially is much higher in responders than it is in non-responders. So what is cluster 3? Oh, so first I'll show just one more thing. Cluster 3 is associated with greater progression-free survival, but also with overall survival for patients. So the more you have of cluster 3, the more likely you are to survive. And this is pre-therapy. This is without any therapy. Patient comes in. If they have cluster 3, they're much more likely to respond to immunotherapy and to have much longer survival. So what is subcluster 3? Well, what we found was that it was composed, interestingly, of the TCF7 positive, CD8 positive cells that are PD-1 positive. And so you can see here subcluster 3, this is the percentage of subcluster 3 containing these TCF7 positive cells. It also contains, cluster 3 also contains interferon gamma positive cells, the ones we had found before. But this was very interesting to us because TCF7 positive cells are really the stem cells of the T cells. And we had shown earlier that the more of these you have, the more likely you are to respond to immunotherapy. And there have been a whole set of studies in mouse showing that these TCF7 positive T cells are both necessary and sufficient for an immune response. So it was interesting to see that subcluster 3, which is the best predictor of outcome in pre-therapy samples, contains these stem-like T cells that can provide a source of T cells for the immunotherapy to work. Because without these, the immunotherapy would run out of T cells quickly because they wouldn't regenerate. And so these TCF7 positive T cells are special in that they allow that regeneration. And so here is just an image again of an example of subcluster 3, showing the markers that I mentioned earlier involved in the formation of this hub. But when we look now at TCF7, which is a nuclear transcription factor, you can see here in red that this transcription factor is present in many of the cells that are T cells based on CD8 in this tumor. So in this subcluster 3 within the tumors. And again, this exists in the responders, not so much in the non-responders. The T cells, the TCF7 positive T cells are further found to be co-present with CCL19 and CCL22. Interestingly, CCL19 is a chemokine, and we'll talk about that more in a minute, that is an attractor of these TCF7 positive T cells through CCR7 chemokine receptor that's expressed on particularly TCF7 positive T cells. So we think essentially what's happening is that these hubs are forming with TCF7 positive T cells due to the presence of a chemokine that organizes these. In contrast, let's say, to the CXCL1011, which is bringing in the activated T cells. So we think that essentially the activated T cells are involved in killing the malignant cells, and there's a positive feedback loop between them and the malignant and myeloid cells. But at the same time, there forms a subcluster of this immunity hub that also contains this TCF7 stem-like T cells that then provide a source for these cells so that you can regenerate and produce more of them as immunotherapy is provided to activate the T cells. So we also tried to make a very simple panel that would capture both of these hubs, the kind of more activated hub and the more stem hub. And we came up with a very simple solution, which is just to stain for the RNA of CXCL1011 and CCL19. And that would allow you to then observe the organizers of those two hubs. Rather than look at everything in the hub, if these are present, the hub will organize and will be present there. And that's what we found. And so this now allows us to do a two-color stain that can be used along in pathology studies by pathologists. So it's not a complex multi-color stain, just two colors, and can be done with standard approaches, but with RNA in situ hybridization. And so we're now applying that across samples. So the way we ultimately defined these hubs is to say we have this activated CXCL10 hub, which is not a stem immunity hub. It contains only CXCL10. We found these stem hubs that tend to be, and these immunity hubs are right in the center of the tumor where the tumor needs to be killed. The stem hub, which also contains CCL19, which brings in the TCF7 positive T cells. The stem hubs tend to be on the edge of the inside of the tumor. And finally, the tumor, basically the tertiary lymphoid structures that people have talked about for a while that are also associated with better outcome, these are present actually right outside the tumor and don't contain the CXCL10 component. So they really contain primarily CCL19, which brings in the T cell, the stems-like T cells. So we think that there are these three structures that interact with each other to provide the spatially organized interactions that are needed to produce an effective immune response to kill a tumor. We were able to look at expression of transcriptome here using the geomics approach, where we looked at particular regions that express these different chemokines. And you can see if we look for the CCL19, CXCL10 positive versus CCL19 structures alone, you can see that these have all the ISGs, while these have, for example, B cell markers such as MS4A1, because these tertiary lymphoid structures are a combination of T cells and B cells. So this kind of confirms the difference between these structures transcriptionally by looking at regions of the tumor. And so one of the most important things we want to do is to get a higher resolution spatial analysis. And so here we developed a 479 gene panel together with Vizgen using the MERFISH technology. And these 479 genes are completely custom and based on the genes we found in the cell types that I didn't mention all the cell types, but all the cell types that we identified by clustering initially, all the gene programs that we identified through the NMF approach that give you all the functional gene programs. This combination of clusters and gene programs allowed us to create a panel that can report on the presence and activity of these cell types and these programs. And so this is a kind of principled way to develop a panel in a kind of unbiased way that captures the natural variability in cell types and programs across a tumor. And so this panel really allowed us to dig in much deeper into these samples. And we worked on a set of non-small cell lung cancer samples, continuing the study I just showed you of the responder, non-responder, imaged with the Murphy system, a collaboration with VizGen of these 479 genes, and did two types of analyses. First, an analysis of tiles that were 2,500 microns squared, or 50 by 50 microns essentially, but hexagonal tiles, to see what kind of neighborhoods are there throughout the tumor. This is a quick way to find out what neighborhoods exist. And then the second is we segmented cells and identified individual cells, and then used those 479 genes to cluster them into subtypes to find the gene programs, and then try to understand in an integrated way what's going on in each tile and each region of the tumor, what cell types are present, what programs are present, and what it tells us about the spatial hubs that I just told you about. So the first thing is we were able, by clustering the tiles across the thousands and thousands of tiles across the tumor, identify these 13 or 14 distinct tiles with different, essentially, gene expression in each tile. And amazingly, we found that tile number 7 expressed CCL9, 10, 11, and CCL19. This is a completely unbiased analysis. You can see 7 sticking out here is slightly different from the others. And that's the one that expresses the two sets of markers that we've shown mark the stem cell immunity hub. And when we look at tile 7 to see what genes are present, you can see CCR7, which I mentioned marks the T cells that allows them to come in response to CCL19. We see TCF7, which is the marker of the stem-like T cell. We see CCL19, which is the attractor of these T cells. So this tile seems to contain the markers that we had identified through the single-cell RNA-seq and the programs and the RNA in situ hybridization that I've shown you, and came out really without any effort in terms of looking for it. It just popped right out of the unbiased analysis. Of course, there were other interesting hubs. The immunity hub itself appears to be T4, where you get CXCL10 and 11, as I mentioned earlier, are the ones that organize that hub. And we also see markers of macrophages and ISG markers and other relevant markers there as well. So we can now dig in and understand both the stem immunity hub and the regular immunity hub without the stem cells, the two that I talked about earlier. And so we can visually now look at this. And what you see is that the stem immunity hubs tend to be on the edge of the tumor, whereas the immunity hubs are scattered throughout the center of the tumor, as I mentioned earlier. And we believe that that's probably a biologically critical point because, again, the immunity hub is involved in the killing through this positive feedback loop that brings all the T cells in, killing of the tumor, and these stem immunity hubs provide the source of T cells needed to kill the tumor. And when we look more deeply at these hubs again, at the gene expression, I already mentioned some of the genes we find in the stem immunity hub. We also find a whole set of chemokines in the stem immunity hub that probably organize the entire set of immune cells that are needed to make this hub function and protect and stimulate the stem-like T cells. We also find IL-15, which is a critical cytokine to keep memory and stem-like T cells alive. So we believe this stem immunity hub really has all the components needed to create a place for the stem-like T cells to survive and have all the cells they need around them to activate them. So now that's at the tile level. If we now cluster all the cells using a combination of CellPose and Bazor to analyze, to segment and identify transcripts within each cell, and then we cluster the cells that we segmented, we find most of the cell types what we actually identified in our original studies of subtypes of immune cells by single-cell RNA-seq, the 479 genes are allowing us to see most of these clusters, and you can see multiple markers per cluster that the original genes that we had pulled out for that purpose. And so now we have a clustering of all the cell types. And now we can ask the question, what cell types are enriched in the stem immunity hub? And what we find, for example, is the CXCL13 positive T cell, the TCF7 positive T cells are enriched in these stem cell immunity hubs, exactly the cells that we were discussing earlier. And so, so we think that basically these stem immunity hubs contain not only the cells we talked about, but also now we can discover other cells. For example, we found that MREG-DCs, which are these dendritic cells present in tumors, originally identified by Miriam Murad and others, are also present in this stem immunity hub, as are CD4 T cells, as are some B cells, and regulatory T cells. And of course, this can be explained by the presence of these chemokines that attract these cells. And so which cells interact? And that's the ultimate question we want to use spatial analysis for. And so we looked at which cells are close to each other, and we used a permutation approach to randomize the positions of cells and asked what's the likelihood that those interactions or distances between cells are not expected by chance. And if that was the case, if we did thousands of random permutations and didn't see that interaction, we can get a p-value basically from that. And that allowed us to identify basically which cell types interact with each other. And so what we found was that the CD8 T cells interact primarily with the CXCL10 positive macrophages. And then the CD4 T cells are interacting with the MREG DCs. And so we think that essentially that there's a network forming both in the stem immunity hub and the immunity hub that essentially explains the physical interactions that bring cells to each other and also coordinate the programs between the cells. And what we've done here is essentially gone from inference from single-cell RNA-seq to actually seeing the cells next to each other and being able to make a hypothesis as to what the functions are. And to summarize what we've found so far, we find that these dendritic cells are present in the stem immunity hubs and most likely are involved in interacting with T cells, particularly CD4 T cells, and CCL19 positive cells, which we haven't talked about today. I won't have time to explain, as another cell type that brings in both the CD8 positive, TCF7 positive cells, as well as the LAMP3 DCs, because they're all expressing CCR7. And that the way the system would work is that the antigen from the tumor would enter these stem-like hubs. The dendritic cells would present antigen, and the macrophages would present antigen, and they would activate the T cells. The T cell would then leave this hub, which is right adjacent to the tumor, and then form a positive feedback loop to be attracted toward the malignant cells until this area of malignant cells is eliminated and there's no more signal, and then the cells would move on to the next area. But the beauty here is that if these cells become exhausted or die, there continues to be the stem cell like T cells that are tumor reactive waiting in the wings for more antigen to come in. And then when the tumor is eliminated and there's no more antigen, these will break up because there won't be any more signal to activate these cells and maintain this hub. And we think that this is analogous to how lymph nodes work, in that there are these regions in the lymph node that contain these stem-like T cells and regions that contain the activated T cells, and they're separated by small distance. And we think that this is actually very analogous to what we see in the tumor. So it could be that the natural systems that the immune system evolved to create these structures are really not only present in lymph nodes, but also directly in the cancer. So I want to finally thank the lung team and the colorectal cancer team that worked on this. John Chen in particular is a pathologist who has brilliantly put together all these studies that we heard today, working together with Linda Neiman on the imaging. Justin Gaynor, who's a thoracic oncologist, who's worked with us closely on all these studies. And Ilya Krasansky, who is a computational biologist who has been working with us closely on the spatial analysis that we talked about today, and Karen Pelka and Matan Hoffrey and Aviv Regev, who we worked with on the initial story I told you about the colorectal cancer. So thank you everyone for listening, and I'm happy to take questions about these hubs and how they form, what their roles are, and discuss further. Thank you.
Thank you, Nir, for your insights. We've received a number of questions from audience members, so we'll try to get through as many as we can in the remaining time. Please be assured that any questions that are not answered live today will be answered via email. So, the first question: are there enough immunity and stem immunity hubs for detection using a standard tumor core biopsy?
Yeah, I think it's important. One question pathologists often ask me is the biopsy actually a sufficient size to see these hubs? And actually, these studies were done with core biopsies. And so we believe that actually most tumors, at least in the lung, where we've been looking a lot, will actually contain enough of them to count them up and analyze them for predictive purposes ultimately.
Thank you. Next question. What therapies would be candidates for inducing more immunity hubs prior to anti-PD-1 therapy?
Right. So one of the big questions here is, these hubs predict, the presence of these immunity hubs, particularly the stem immunity hubs, predict response to anti-PD-1. So ideally, one would want to induce more of them before you give anti-PD-1 therapy. So, clearly inducing more antigen-specific immune responses, such as with vaccines or in situ vaccination, could be an interesting way. The other is to try to induce some of the markers that allow, or the genes that allow TCF7 positive stem cells to survive. And so there are several ideas out there right now on how to do that. For example, JAK inhibitors have been used recently to reduce the immune response in tumors after anti-PD-1 therapy to non-responders, and that may allow less exhaustion and more of these TCF7 positive T cells to expand, so that you come back with anti-PD-1 therapy and you get a larger response. And then, of course, the BRAF/MEK, which I mentioned today in the talk, is one way to induce the chemokines that actually are one of the major organizers of the immunity hub. So we think there could be a lot of directions to go and trials that may be interesting for this direct purpose.
Thank you. And what is the difference between TCF7 plus T cells in the tumor hub versus those in draining lymph nodes?
Yeah, so there are a lot of TCF positive. TCF7 is a transcription factor that marks basically long-term surviving memory cells in T cells. And so there are memory cells in the lymph nodes. Those are more primitive memory cells that can survive for many years. And we believe the difference is that the ones in the tumor are not all these long-term surviving memory cells, but are precursors of exhausted cells, which also have TCF7 and also are self-renewing. So they may be a reservoir of cells at the tissue site that allows responses to occur rapidly, but they may not have the same survival potential as the ones in the lymph nodes.
Great. Is it possible to study immune hub by using IHCs only? If so, which antibodies should be included?
Yeah, so we proposed, and I mentioned it in the talk, that actually since the two key chemokines that we've identified, CXCL10 and CCL19, are the ones that organize these immune hubs, a stain for those two will mark pretty well the existence of these hubs. And so I think that could be a very good way. We're doing IHC now with those two as RNA probes, not as antibody probes, but it's very feasible to do that as well. So I think that's a way to mark these hubs that I talked about today in a two-color stain.
Thank you. So next question, what about the bioinformatic analysis of RNA sequencing and the maximum benefits from output data from RNA sequencing?
Yeah, so RNA sequencing, single-cell RNA sequencing was a key element of our analyses here. Without it, it would have been very difficult to create the spatial panels that we talked about today. So the almost 500 gene panel that we created using MERFISH was possible because we were able to analyze with single-cell RNA-seq in an unbiased way, genome-wide transcriptomes, what the differences are between the different cell types in the tumor and what gene programs get turned on in the tumor, particularly in responsive versus non-responsive tumors. So by being able to identify all those genes prior in the single-cell RNA-seq, and you can see that in our paper we had a couple of years ago, Pelka et al., how we do that. By identifying all those genes, then that allows us to create a panel for spatial analysis to dig deeper into the key cell types and programs at a spatial level.
Fantastic. And the next question, when do you think spatial analysis will make it to the bedside in clinical practice? And if so, do you anticipate it will be only in specialized labs, or could it be simplified enough to be in the hands of local community hospital pathologists?
Yeah, that's a good question. I mean, certainly the highly multiplexed one, I think, will be relatively difficult both for interpretation as well as for quality control at this point. It's really a very good research tool, but it's a good discovery tool for identifying the key, key stains and spatial organization that you would need for a more standard pathology. So I think that it's more a discovery tool at the moment. I think it will take a bunch of years to get it to the point of reproducibility for regular use by itself.
Okay, great. And have you done TCR sequencing for this experiment? If so, did you do any TCR clustering based on editing distance that correlated to these subclusters?
We've started to do some TCR analysis. It's pretty difficult to do spatial TCR analysis. We've done a little bit, suggests that the structures are related to each other in the sense that this STEM immunity hub contains some TCRs that are shared with the immunity hub within the malignant cell area. So we think that there's basically, and by single-cell RNA-seq, we have done a lot of TCR analysis. And so there's essentially clones that develop all the way from the stem cell stage to the effector and exhausted stage within the tumor that we're looking at. But we haven't yet done really careful single-cell level spatial TCR. We're about to do that.
Okay, great. And CD4 Th1 cells are also known to express CXCR3 receptor for CXCL9, 10, 11, potentially contributing to IFNG secretion. Have you checked if this population is enriched in these immunity hubs?
Yeah, they are. The CD4 T cells that we see enriched in this hub are generally CXCR3 1 positive. So yes, absolutely. And so that's very likely why they all find themselves in the same hub.
Okay, fantastic. Unfortunately, that's all we have time for today. Again, let me reassure you that any questions left unanswered will be answered after the event by email. This webinar will also be available on demand in just a few days, and you'll receive an email alert when it's ready. Thank you once again to Nir for presenting, and thanks to everyone for joining us today. Hope to see you all in the future at different webinars. So goodbye for now.
Thank you, everyone.